sorry that we haven't had a break, but I'm very happy to be here. So my name is Dr. Hannah Ray. I'm a clinical psychologist coming from the RAB lab, which is the Research in the Autism and the Brain Lab at the University of Washington. So people have been throwing around the word tiger study, so I'm here to talk about that one. I am a clinical psychologist, so I am going to attempt to talk about genetics, but I am not a geneticist, but I'm going to do my best here. The other thing is that my specialty has really been in autism, but I'm very excited to say I'm moving into Down syndrome research, so I'm like particularly excited to be here and meet you all. All right. So goals for today are to talk about our study, the Tiger study, and a little bit more about our lab and our approach to things. From what I have heard, I'm sorry I couldn't be here yesterday, but the flight from Seattle is long. So I don't think you guys have really talked that much about like what are genetic events generally and DYRK1A kind of more specifically. So I will talk about that, but you can also like wave your hands and be like, we're good, we don't need to hear this, and I will not be offended. And then I'll talk about some of the research findings that we have had coming out of our Tiger study. Okay, so the investigation of genetic exome research, or the Tiger study. So like I said, I'm here to talk on behalf of the RAB lab. So our current leaders of the lab are Emily Newhouse, she's also a clinical psychologist, and Evan Eichler, who's a geneticist. We also have had a lot of really amazing researchers who have really built up the lab. So I want to call out Caitlin Hudak, Eva Kurtz-Nelson, Raphael Bernier, who really started the lab, and Rachel Earle. And then we also have had so many other research collaborators and other staff members who have really been important to our lab. And really our goal in our lab is to make research accessible and applicable for our families. So each person's research contribution we know is really essential in informing our understanding of neurodevelopmental differences. And this helps us to understand and identify innovative ways to improve care for individuals and families. And it is just so cool to hear how interested you guys are in research, and really it's just like, as a researcher, it's often so hard to get participants to join us. And so it is so cool that you guys are so excited about it, because what we really want to do is, sorry, hold on one second. We want to enhance our understanding of neurodevelopmental differences using a lot of different methods. So our lab is very much focused on autism, but we're really looking to identify clinical subtypes of autism associated with specific genetic variants using a genetics first approach. And so what is a genetics first approach? So we include individuals because they have a certain genetic event that is associated with autism, but we include people who do not have an autism diagnosis. One second. I think I sent the wrong version. One sec. I sent you the wrong version of the PowerPoint. I am so sorry.
You need to join Meet. Can I send you an email?
Sorry, give me 1 second. Good time for the break that you didn't get. Do you want me to resend it? Would that be easier?
It could be
Here. I'm so sorry.
No worries.
Do you want me to send it to you again?
Yeah.
Okay. Admin at DYRK1A.
That works.
Okay. Should be this one. Oh, you know what? It didn't update. It's weird. I see the problem. Please, I'm so sorry. Okay, that should work. Sorry, I had old slides up here, and my new ones are better, I promise.
Or like I'm doing.
I can talk in the meantime. So, well, let's talk a little bit more about what is autism and kind of like what it should look like generally. So autism is defined based on two categories of behaviors that we're going to show in a second. So these behaviors kind of fall into what we call like two buckets. So we have social communication differences, and then we have restricted repetitive behaviors and interests. So social communication differences include decreased reciprocity. So maybe someone has trouble going back and forth in a conversation. We have reduced nonverbal communication, so that can be like less use of gestures, less eye contact. And then we have challenges in relationships. So some kids are super interested in relationships, they just have trouble forming them, and other people are less interested in relationships at all. Then the restricted repetitive behaviors and interests, which we call RRBs, those include repetitive behaviors. So kids with DYRK1A, we see a lot of hand flapping and arm flapping and things like that. And also repetitive use of objects. Then there's the insistence of sameness. So a lot of kids like routine, that's one example. Then we have restricted interests. So some kids have interests that are kind of unusual in what they're interested in. Other kids have really intense interests. And then we have sensory differences. So those can be interest in sensory experiences. I think that's what we see a lot in DYRK1A. So for example, a lot of kids with DYRK1A are really sensory seeking. They really like lights. They really like loud noises, things like that. And then we also see kids with sensory aversions. So in order to have a diagnosis of autism, you have to meet all of the social communication criteria and then have like 2 of the RRBs. And like I just said, there are a lot of examples in all these categories. So kids with autism present very differently. And so one of the things that we talk about with the genetics first approach is, can we understand kind of why these kids are presenting differently? Can we understand how differences are similar, or similarities and difference between kids with different genetic events and kids without genetic events too?
Don't need to worry.
Okay.
He's here.
Thank you. I'm so sorry.
This one, right?
Yeah. So those are the categories, like I said. Thank you so much.
No problem. It's the clicker.
Okay.
For this. Oh, what's right here?
Yes. And now we can talk about what the genetics first approach is. You can just click.
Yeah.
Yeah, okay, excellent. All right, thank you. Okay, so genetics first approach, genetic variants that relate to autism. So we have known since the 1970s that genes are contributing to autism because we know that autism runs in families. We used to think that there would be an autism gene that we could find. So that would be like one consistent change to a gene that would happen in everyone with autism. So based on that expectation, researchers took a phenotype first approach, which is kind of this. Can you see my mouse? Yeah. Okay. This side. And so what they did was they got a lot of people with autism, and then they did genetic testing on those people looking for a common genetic event. So researchers were able to find a lot of genes that were associated with autism using that approach, but they also found that plenty of people with the same genetic event didn't have the same diagnosis, and that there were a lot of different genetic variants that related to autism. That's really tricky because for research, we like to have a lot of people. A lot of what it seems like you guys have been talking about today is like samples are small. So one way that we can increase sample sizes is to use this genetics first approach, where we get a lot of people with the same genetic event, and then we try to look for similarities in clinical diagnoses between people with the same genetic event, differences, and also differences between people with different genetic events. So for TIGER specifically, we've gotten funding 3 times from the National Institute of Mental Health to take this genetics first approach. And that way we can study clinical subtypes of autism that are associated with specific genetic events. And one of those subtypes is DYRK1A. So as you can see in this flyer, we are inviting families with genetic events variants to participate. That list does change. Evan Eichler is in charge of deciding which genes we're studying. This is the current list of genes, and you'll see that DYRK1A is one of them. So we enroll individuals 4 years and older along with their families. We need at least one parent to be fluent in English. Dr. Taylor talked nicely about we are actually now able to include international families thanks to COVID. But some of the assessments we can do with non-English speaking families, some of them just aren't standardized in those populations, so we can't quite do that. On the next slide, I'll talk more about the data we collect. We do collect very different data than a lot of the other people I've seen speak today. So there is kind of overlap, but you do often have to start from scratch a little bit with us. And we are also, what's kind of unique about us is that we're collecting data on the kid with the genetic event. We're also collecting data on biological parents who are interested in participating. And that's because we really want to understand how parent genotype and phenotype might contribute to the child's phenotype. And so like Dr. Taylor talked about, it is harder to find individuals who are older, but with parents, I think we have identified at least 2 inherited genetic events. So we found parents with the gene event of for DYRK1A specifically. We also accept kids who are adopted. So in that case, the parents wouldn't fill out information about themselves, only talk about the kid. We cover costs of participation. We do collect saliva for genetic testing. So we would pay for the saliva kit and the shipping and everything like that. For families who don't want to do saliva, we also offer blood draws. We did used to fly participants out to Seattle to participate for 2 days. COVID obviously kind of ended that. But it has actually been really nice. We've been able to adapt pretty much everything to be on Zoom or on the phone. And that is actually really accessible to a lot of families who might not have been able to take 2 days off work and fly their kid across the country. And so like we have very flexible hours with our calls. It's kind of nice that we're on the West Coast when our families are on different time zones, because what's late for you, like after work for you, is middle of the day for us. And so we found that we've been able to get a ton of really helpful information and to be really a lot more flexible with families who had a harder time participating before. And then to participate, I'll have this again at the end, but you can contact Connor, or if you just want more information. All right, so here's a little infographic about the type of different activities that mom, dad, and the kid complete. It is a lot more intensive than the other studies I've seen talked about today, so it is 15 to 20 hours spread out through all the individuals. I know that sounds like a lot, but we are able to really dig into the diagnoses and the phenotype a lot more. So for example, when you're diagnosing autism, the way to do it, like the absolute best practice, is that you have a really detailed developmental history in addition to seeing the current presentation. And so we are able to do all of those things, and that takes time. Initial items that are shown here include calls with Connor. He'll talk to you about the study, they provide consent. He will ask for a gene report. If someone's having trouble obtaining a gene report or doesn't have it, you can give him permits. The participant can give him permission to obtain it. And then for the cognitive behavioral section, we have questionnaires, we have interviews, we have behavioral assessments on problem solving, autism traits, mental health history, and the child's intervention history. And so even though our lab is focused on autism, we also know that there are a lot of co-occurring conditions that are really important to these genetic variants. And so we do our best to capture not just autism, but other co-occurring conditions that are relevant. We know that not all parents can have a phone call with us. So for example, like it's hard, sometimes mom can participate, dad can't, that's totally fine. So for our international families, they can't necessarily call us on the phone, so we do Zoom calls. We know a lot of parents are really concerned about their kids' ability to sit still for a Zoom assessment. First, we found that like pretty much everybody can do it because we make them really short on purpose. And if a kid is really having a hard time, we have interviews that we can do instead. But it is really fun to get to meet the kids, so we appreciate when parents encourage their participation. The medical items include, we teach parents how to measure the kid's head circumference, weight. They take some pictures of the kids. They answer questions about the kid's medical history, and then there's the genetic or blood sample, or sorry, saliva or blood sample. Again, everything is optional, but this is kind of ideally what we're able to get. And then the final items I'll talk about at the right now. So we do have a feedback session, and in that feedback session, we're telling families what we found about the kid. So diagnoses, recommendations for them for what kind of care might be helpful, things like, and also we talk about the other kids with DYRK1A that we've seen. So I'm going to talk to you today about the kids with DYRK1A that we've had so far, but as we are continuing to collect data, that changes. Families also get $100 for participating, and then obviously it contributes to lovely events like this, and to the more information we're able to get, the more we can improve medical care for kids with DYRK1A. Any questions about Tiger before? Yeah. 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 Yeah, so that's a really good question. She asked if we update families. First of all, so we can't diagnose on Zoom, but we have been able to give parents a little bit more clarity about because autism in kids with rare genetic disorders is not particularly well understood by clinicians in normal clinics. So it's not surprising that it can be really hard to get a diagnosis. But we are specially trained in that, so we can maybe say, we can't give you a diagnosis, but here are the features that we're seeing that would suggest autism. A lot of times we do newsletters. I will see if we're up to date on that so that we can give parents more updated information. We do try to come to this family meeting every year to give you guys updated information. What other kinds of things would you like to see?
Well, I mean, I think if there's types of therapies that you're finding that might be working in this population, or what's your mark, especially, I mean, let's focus on autism. We actually went out to see you guys.
Yeah.
But I mean, there's probably other stuff that I
Yeah.
don't know.
Yeah, so we are in the process of pulling together a paper about intervention usage in this in all of the kids that we've seen, and I'm going to talk about that data a little bit today. What I'm going to talk about today is not like family satisfaction or like how effective it was, because we don't really have that information. So what we do is we ask parents, what therapies have you enrolled your child in? How satisfied are you with them? Things like that. So I'm going to talk a little bit about the trends that we've seen and what people are accessing. I think because we don't know, we can't study treatment outcomes unless we were kind of doing the treatment and getting the information, but we will work on continuing to find, we always definitely want feedback on the kinds of information that's useful to you guys, because that is a huge point of this. So that's good to know that that's something you're looking for. And something I'm not talking about today, but I can probably rummage together if you need, is we have families engage in a lot of different kinds of therapy. And so I have a list of those somewhere on my computer that I can find, just in case you kind of need ideas. Other questions about the study? Okay. So we'll talk a little bit about DYRK1A and about genes. So first, let's talk a little bit about genes, genetic variants, and what they are. So DNA is your body's instruction manual. It has information on how to build you and maintain you. I'm going to go very basic, just to make sure we're all on the same page. So we think of genes kind of as the paragraphs or the short segments that make up the DNA. And then one molecule of DNA with one protein make up one chromosome. So I think of it like a library. Yesterday I read it's like genes are a house in a neighborhood, and DNA is the neighborhood, and chromosomes are more neighborhoods or something. So in your body, you have 46 total chromosomes that are organized into 23 pairs. These chromosomes are in each of your body cells. And in order to get all the cells that you need for your body. You start with a few cells that divide, and then when these cells divide, they handwrite a copy of your original chromosome word for word, which sounds tedious and silly, but here we are. And then you end up with about 25,000 genes in your body. So you can imagine with handwriting all of those copies, you're going to have mistakes and changes. It happens in pretty much like in a lot of people. I've heard the term de novo thrown around. So some of these changes are brand new changes or events. They are called de novo events, and they're not inherited by either parent. They're not caused by anything anyone did. They just happen. And the likelihood of a mistake or a change happening in another offspring that the parent has is less than 1%. Then there are also inherited changes, and those do come from the mother or the father. So if one parent has a genetic event, there's a 50% chance the other kid will get it. That said, we have seen that some mothers or fathers might have the genetic event, but might not have the syndrome. We don't know why any of this is. We don't know why a genetic event causes a syndrome in some people and not in others. That's the point of research, is to figure these things out. All right. So I just said genetic events happen a lot, and there are a lot of different kinds of genetic events, which a lot of people I've seen today kind of introduce. We'll talk about those just quickly. So genes are made up of a series of letters. So this is the original sequence, you can see these letters can be changed in different ways, and that's called a point mutation. So for example, if the original sentence or the original sequence was the gray cat ran down the hall, missense would be changing the letter hall to ball, and it just kind of changes what the sentence means. A nonsense would be you change something about the sentence and it causes it to stop creating the protein. And then we also have insertions, deletions, or duplications. So as you can see with those, it kind of moves everything around. And so these changes can do absolutely nothing. They can change what the protein is like, what proteins, like how the protein's functioning, or it can just cause it to stop creating a protein. All right, so now that we have some very clinical sense of what genes are, we can talk about DYRK1A a little bit more. So it's located on chromosome 21. So I've heard it talked about in reference to Down syndrome. That's the Down syndrome critical region. So DYRK1A gives instructions to make a kinase, and this is an enzyme that speeds up chemical reactions that are important in the development of the nervous system. It's important, so DYRK1A is important for making brain cells. It fixes connections between brain cells. It impacts development starting before birth and in infancy. So that's why I would think it's so correlated with like intellectual disability and autism, these brain changes. DYRK1A syndrome is happening in less than 0.5% of the population, which is why it is so hard to study. But thank you all for contributing to research on it. And we call it DYRK1A syndrome when there's a haploinsufficiency, which is an underexpression, such that one copy of the gene isn't making enough of the kinase. And like I think Dr. Taylor talked about, we've seen changes, we've seen additions, we've seen deletions. Everybody seems to have a different version of the genetic change, but there are similarities in the syndrome that we'll talk about. Also, it is rare that DYRK1A is inherited. In the 27 people that in 27 of the people that we've seen, only 2 had confirmed inherited cases. So most of the time it's de novo. All right, and then we talked about Safari today. So Safari has this really cool resource where they list all of the genes that are associated with autism, and they give us a score of how confident we can be that it's associated with autism. So here we see that DYRK1A is 1S. So S means it's syndromic, which means it's associated with conditions other than autism in addition to autism. The 1 means that the gene has been clearly associated with autism. The Eagle scoring, anything greater than a 12 means that we have strong evidence. So this gene has been repeatedly demonstrated to be associated with autism. And I know I've heard a lot kind of like in watching Eva's talks and kind of today, like concern kind of, do people care about DYRK1A? And the strong association with autism and with intellectual disability is kind of encouraging because people are working really hard to understand autism and intellectual disability better. And so because this is so strongly associated, it is often included in studies that are looking at the genetics of autism. All right. So now we're going to talk about the research we've done on DYRK1A syndrome in our lab, and we're going to be talking about co-occurring conditions, which I'll breeze through because Dr. Taylor talked a lot about that. And then we'll talk a little bit about the interventions that we've seen people use and kind of what, just kind of like generally what that's looked like. All right. So we, in a publication that is coming out soon, that's another way to learn about some of the things that we're finding in our lab. And Eva talked about this last year, so I'm not going to go into a ton of detail about it. But basically, we compared the presentation of autism in kids with DYRK1A to kids with autism who don't have genetic event with. Is that me or someone else? Oh, it does that. Great. Thanks, Siri. All right. To kids with low nonverbal IQ, so other kids with intellectual disability, and then kids with autism with no genetic event with average IQ. So in most of the people came in person, so most of the people were from before COVID, but we're still collecting data. We do think we have about a 50-50 female to male split. We think that's pretty typical for DYRK1A. And we matched people based on age range too, pretty much. So like I said, our study does a very detailed assessment of autism. So we have like a 2 to 3 hour interview where we get a ton of developmental information and also current presentation. We have this observation, which we have adapted to be over Zoom. So even though we can't diagnose based on it, we get to interact with the kid and really get a sense of what current presentation we're seeing. And then we have some parent reports too. So if we're comparing the amount of autism traits based on clinician observation between, again, kids with DYRK1A, kids with autism with no genetic event, and with low IQ and average IQ, we're not really seeing differences in the presentation. So that means that the intensity or frequency of clinician observed autism traits are comparable across groups. So there's like slight difference, but really it's not significant. It's pretty similar. It changes when we get to the parent interview a little bit. So for these graphs, we're showing what % of parents are reporting that their kids showed the behaviors that look like autism. And a higher score indicates that you show more intense autism traits. So it's more characteristic of autism. The numbers next to the dots indicate the number on the item number on the assessment, which isn't super meaningful to you, but if you can kind of see the categories that we're talking about up here. So we have nonverbal communication, relationships, and social emotional reciprocity. And so I can talk more about the items if anybody wants to hear about them later, but generally what we found is that kids with DYRK1A are scoring the highest, which again means more autistic traits. They're not significantly different from the kids with autism with intellectual disability. So that means that by parent report in these interviews, they present pretty similarly to other kids with intellectual disability. The only kids that they're significantly higher than are kids with autism with average IQ, average or above. And we see the same thing with these RRB categories, so repetitive motor behaviors, restricted interests, routines, and sensory differences. That generally the kids with DYRK1A look like kids with intellectual disability. All right. The other thing that Dr. Newhouse is currently in the process of working on and kind of looking into more are these co-occurring mental health conditions. So on the right side, you see the lighter colors are the kids who have pretty average scores based on parent report in anxiety, affective problems, which is like depression/anxiety, ADHD, and oppositional behaviors. And the darker colors are the kids with elevated, so like really clinically significant. We're a little bit more concerned, more elevated compared to the general population in those areas. And so what we found in DYRK1A is that there do seem to be relative elevations in affective problems, so that's like anxiety and depression and ADHD, and lower concerns with anxiety and oppositional behavior. And then the other thing about like having small sample sizes, so even though there's not amazing collaboration between different research studies, researchers love to do reviews and to publish based on as many people as possible. So the last review I could find was from 2021, and it was based on 68 individuals. So here you see the percent of the condition based on that review study. And so it's pulling together all the published information that they could find. Then I have the percent of the condition just in Tiger. So we've talked a lot about intellectual disability, that's low IQ plus low daily living skills. We also talk about global developmental delay. So someone said, is the rate of intellectual disability not 100% because the kids are young? Global developmental delay is the diagnosis we give to kids who are younger, who are behind in their developmental ability. So problem solving, motor skills, things like that. So I just was looking this up, which is part of the reason I had to email the new presentation. So our rate of 90% intellectual disability, the other 3 people have average or low average IQ. So it is not 100%, not because the kids are young or anything like that, but we actually know we have found some people with average to low average IQ. For speech impairment, I think the reason that our rates are so much lower is that I am only talking about people with like a diagnosis of like an articulation disorder or other kinds of speech concerns. So like speech sound disorder, which is difficulty with pronunciation, things like that. Most people have microcephaly, feeding problems, eye abnormalities, and seizures. And for seizures, I'm grouping together febrile seizures and other seizure diagnoses. And then as I kind of just talked about, so we actually have a higher rate of autism in our study. It's a little bit hard to know why we've seen estimates between 45 to 86%. Some of that might be because we do such deep phenotyping on autism. And the other thing that I'm sure you guys all know better than anyone is that kids don't necessarily have an autism diagnosis, but they have a lot of autism traits in kids with DYRK1A. And then, like 11 to 27% of kids are having anxiety and about 7% are having ADHD. Any questions about the co-occurring conditions or autism in DYRK1A? So now I'm going to talk a little bit about the data that we're in the process of looking at on interventions. So we have the percent of kids enrolled in intervention. So you guys are doing a phenomenal job getting your kids into speech therapy and occupational therapy. Just hats off to you. Behavior therapy is definitely utilized a lot less. One of the things I'm wondering is if that's because it is a little bit harder to get an autism diagnosis, and so sometimes it can be hard to qualify for behavior therapy if you don't have that diagnosis. What? Yes, and even if you're diagnosed, you can't get the services. So one of the things that I work on very heavily are parent training programs for behavior, because like things like ABA, while first of all, not everyone loves them, they also take 20 to 40 hours per week, and so then there aren't that many providers to do that. So there have been adaptations to behavioral programs so that you can instead teach the parent for an hour a week, and then the parent can implement the skills. So it's a lot to ask the parent, but you can probably actually get in. So this is any parent who said that they were getting behavior therapy. No, it's not only ABA. It's other, I would have to look at what other therapies parents listed. I think it's often ABA, and I think, so. So the other thing that we, the other data that I wasn't able to pull together for this, but that so parents for our study list out every intervention that their kid has pretty much like ever been in. So the best way we could find to group it is speech therapy, occupational therapy, behavior therapy. Then we have an other evidence-based therapy category, and then we have an alternative therapy category. And so I wasn't really able to pull the numbers on those because like there are so many different kinds of therapies that families are enrolling their kids in. In the manuscript that we're coming out with, that looks at not just DYRK1A, but like all of the genetic events that we are collecting information on, we found that kids with, we did find a lot of alternative therapy use, and it was related to autism traits, I think. I have to like go back and look at this because we're deep in it right now. And again, that was kind of part of where I came up with this idea of maybe kids without the diagnosis are enrolling in more alternative treatments, because that's what they're able to get into. I'm not really sure. Yeah. Physical therapy would be separate, and I don't think we had high rates of that, unless.
I mean, like Grayson had told.
Yeah, so it's based on what parents called it. So that's where messy gets a little bit data gets a little bit messy, sorry. Is that it's like, you know, the school might call it occupational therapy, but it's really physical therapy, and so parents are reporting on that, or parents are trying to remember what their kid is in. So this is what parents called occupational therapy. Okay. So then I just wanted to talk a little bit about like how long is everybody spending there? So you see here that this is the weeks per year. So families are spending a lot of time there's like a really wide range in how long families are spending in occupational therapy. My guess is that's because some people are getting it in school, but I'm not 100% sure. You guys could tell me about that. And then generally parents are spending it like, there's less variability in how much time people are spending in speech therapy. So the reason behavior therapy is weird, and the reason I think a lot of it is ABA is because like there were like, I think 4 families who were able to get behavioral therapy, and the ones who are in it for like most of the year. So that makes me think it's probably mostly ABA. And I think those other interventions you were talking about would probably go into the other evidence-based therapies, because things like floor time and other autism interventions aren't necessarily targeting behavior. And we're talking more about therapies that are targeting challenging behaviors. And then we also just get like a huge range of how long per week families are spending in therapy. And this is just kind of generally like how many hours per week. All right, so some takeaways. So most kids with DYRK1A syndrome have autism and intellectual disability, with a huge range in rates varying across studies. It is actually really great, I think, that there are similarities in autism presentation in kids with DYRK1A compared to kids with autism with intellectual disability, because we would think that would mean that intervention shouldn't have to be tailored too much. Like generally implemented intervention should probably work pretty well for your kids. Because the presentations are similar, and like all autism interventions are tailored to the specific presentation of that kid, because kids with autism already vary so much. So my guess is that intervention should work pretty well. It's always a matter of finding providers who can take you, but I think that in general, we would expect similarities in outcomes. Great. And then just if anyone is interested in participating in our research or has ideas of other things that you guys want answered, you can talk to me. Connor is a research coordinator. All right, and so Caitlin Hudak, who I mentioned at the very beginning, her interest is really in EEG and understanding neural functioning in kids with rare genetic disorders. She said offhand recently that she's working on getting a study started up on DYRK1A. So I don't see it on her website yet, but you can email her if you're interested and ask. I don't know if she's going to do this again, but because of COVID, she did a cross-country road trip and did EEG in everyone's houses, which is like incredibly ambitious. And she has very cool videos and is really talented at collecting EEG data for kids with rare genetic disorders. So I would stay tuned because her research is great and she came from our lab, and really we collaborate really closely with her. So again, thank you guys so much for participating in our research. I'm happy to take your question, and also just for being here and for letting us come talk to you guys and get to talk to you about what we're doing.